Next up, we have Dave Lovinger from National Institute on Alcohol Abuse and Alcoholism. He's going to talk about dopamine, dope, and sleep. You have 20 minutes, Dave. You're muted, Dave.
Yeah, I definitely enjoyed the last talk, and thanks everybody for inviting me. I am going to talk about dopamine and dope quite a bit, dope being cannabis. I will go a little short on the sleep data because it doesn't line up as well with the dopamine findings we have, but I will try to mention that at the end of the talk, and we discuss it more if you like. So what has stimulated our interest in endocannabinoids, cannabinoid drugs, and dopamine is kind of schematized in this slide here. And what we're showing is that summaries of studies, mainly from Joe Cheer and Carl Lupica and others, that modulation of activity in the midbrain by these lipid-derived endocannabinoids that can signal retrogradely to the CB1 receptors on presynaptic terminals inhibit transmission onto these midbrain neurons. And in fact, the inhibition of this GABAergic transmission disinhibits the dopaminergic neurons and increases dopamine levels. Conversely, in the nucleus accumbens, CB1 has a much more role in a much more complicated regulation of dopamine release, whereby CB1 receptors activated by endocannabinoids on the terminals from the prefrontal cortex that release glutamate leads to a decrease in ultimately in dopamine release. And this is kind of a complicated paradigm that Yolanda Mateo has shown, that it also involves decreased acetylcholine release. So I'll leave you to read about that. But the bottom line of these studies is that endocannabinoids, through a bunch of mechanisms, can modulate dopaminergic function. And Joe Cheer's lab and others have also found that endocannabinoids contribute in vivo to dopamine increases induced by a variety of stimuli. Of course, you all know from watching this conference that dopamine increases or decreases can signal reward and reward expectation, and that Joe has found evidence that endocannabinoids play a role in this type of dopamine signaling. But what we're more interested in for the context of this talk is that endocannabinoids are implicated in dopamine increases produced by a variety of drugs of abuse. So I'm showing here Joe's data using fast-scan cyclic voltammetry in the nucleus accumbens in vivo, showing that, of course, as you expect, cocaine treatment produces large increases in dopamine in the accumbens. And ethanol also produces increases in accumbal dopamine. And these are inhibited by rimonabant, this inverse agonist of the CB1 receptor. And so these data indicate that endocannabinoids actually play a role in drug-induced dopamine increases. And in fact, we also know that cannabis drugs can increase accumbal dopamine. So that these backgrounds sort of stimulated our interest in looking at how cannabinoid drugs interact with the dopamine system and how that might affect behaviors. And we're particularly interested in this cannabis use and withdrawal related behaviors. So as you know, cannabis is widely used in the US, probably the most widely used illicit drug in the US. There's currently an effort to legalize it. It's legal in many states, and people are even touting the therapeutic potential of cannabinoid drugs. But we also know that chronic cannabis use can lead to this cannabis use disorder that's now recognized by the American Psychiatric Association. And this involves use despite adverse consequences, craving for cannabinoid drugs during abstinence, and tolerance to the effects of the drugs. And of course, with this as a cannabis withdrawal syndrome that involves these symptoms, things like irritability and other mood changes, nausea, anxiety, decreased appetite, and importantly, sleep disruption. This is something in which we are very interested. So these effects of cannabis drugs largely stem from the actions of delta-9 THC, or tetrahydrocannabinol, shown here. I'll refer to it as THC. This is, of course, the main psychotropic phytocannabinoid found in cannabinoid drugs. It's responsible for the development of cannabinoid use disorder, and its cessation of use invokes these withdrawal symptoms. And it produces its effects as a partial agonist of the CB1 receptor. So we want to model the effects of THC, and especially this THC withdrawal syndrome, or this cannabis withdrawal syndrome. And we'll do this in the mouse by giving them THC. But there are two sort of preferred ways of doing this. One is the spontaneous withdrawal model, in which you give the animal the THC treatment that I'll show you in a bit. This evokes tolerance. You just stop the THC cold, and then you get withdrawal symptoms, but they're generally fairly mild. Whereas with this precipitated withdrawal technique that a variety of labs have used, you do roughly the same thing, the treatment that leads to tolerance, but then you give the animals a CB1 inverse agonist. And this produces admittedly reliable and stronger withdrawal symptoms that are easy to measure. But we don't think it models very well what happens in the human conditions, because they rarely encounter CB1 inverse agonists. So we're going to use this spontaneous withdrawal paradigm. And as you'll see, we can see pretty robust changes in dopamine and interesting changes in behavior with this approach. So here's the approach, along with the person driving the studies, Dr. Andrew Kestner, who is a postdoc in my lab, now become an independent research scholar at NIH, our first. And then with help from very talented postdocs, Alexa Gracias and Stephanie Ramos Maciel. And what they've been doing is this chronic THC paradigm that's designed to produce tolerance. The reason we're using this, it involves a rather high dose of THC, but we know that will induce tolerance to the THC actions. So after a acclimatization period, and the mice will receive, these are C57 black 6 mice, they'll receive either vehicle or THC over this 6-day regimen. And then we'll simply withdraw the THC administration and look in abstinence on day 2 of abstinence, or later on day 6 or 7 of abstinence. And then what I'm going to be showing you first are data that we collected for dopamine measurement in slices from the dorsomedial, dorsolateral, striatum, and the nucleus accumbens. And to do this, of course, we're using the tried and true fast-scan cyclic voltammetry paradigm, in this case spearheaded by Yolanda Mateo, who must have done 1 million of these type of recordings. So this just shows the location where we're putting our carbon fiber electrodes to measure dopamine. We're using the standard ramping protocol to get oxidation and reduction. So we're sure we're looking at dopamine. Then in this case, we're using electrical stimulation in each of the regions with a nearby carbon fiber electrode to measure dopamine transients evoked by single stimulus pulses in the dorsomedial, dorsolateral, and the nucleus accumbens. And so what I'm showing you here first is the data with chronic treatment and withdrawal or abstinence in the female mice, because this has actually turned out to be the more interesting data. So what I'm showing you here are typical traces, the gray, the vehicle, the green, the THC treated animals, and then these input-output curves where we increase stimulus intensity and we see increases in dopamine release. These are once again single pulse stimuli. And what I think it's pretty easy to see is in this early withdrawal period, a couple days after withdrawal, we see a consistent increase in dopamine release in all 3 striatal subregions in these female mice. But by the end of the withdrawal period, we really see no change in dopamine release. So that's kind of interesting. Even more interesting is that the males don't show hardly any change. And so here we're looking at the 3 different subregions. You can see the traces here, these input-output curves in all 3 subregions early and late in withdrawal. And the only time point and place where we see enhanced dopamine release with the THC group is in the dorsomedial striatum late in withdrawal. So there is some differences that we need to dig into about the mechanisms of dopamine release that underlie this enhanced sensitivity of the females. But what we also want to know is during the withdrawal period, could that enhanced dopamine lead to any behavioral changes that might make the condition worse or better for the female mice? So to do that, we performed a large battery of behavioral tests, and Andrew can tell you all about that. But I'm going to focus on one today. But first, I'm going to show you the paradigm that we used. First, we do this acclimatization habituation period. Then we have a period of baseline measurement for all our behavioral tests, where we actually get a behavioral baseline or train the animals to perform the task that I'll show you in a second. And then we have this chronic treatment paradigm, same as I showed you before. And then we're going to be looking at the behavior early in abstinence, the first 2 days of abstinence, and then the last 2 days of abstinence for this late period. And so what I'm going to be showing you here is results from this operant cue discrimination task. And this is designed to test several components of goal-directed behavior and conditioning. So this is a task reminiscent of, I'm sure a lot of people have used. The animal, it's a self-paced task where the animal can press one of these, either of these two levers. And when it presses the lever, there's a 50% chance that there will be CS plus tone delivered, and that will tell the animal that sucrose is available. And then it should go to the port and get the sucrose. On the other hand, 50% of the time there'll be a CS minus presented. Of course, this is all randomized. And then that'll signal the animal that no sucrose is available. And the animal just keeps doing this over and over. We pre-train them to get used to the aspects of the task. Then we pre-test them for 2 days where they go through this paradigm for several trials every day. And what you can see is that the animals become quite good at discriminating the CS plus, both the males in blue and the red females in red. Their approach latency to the, or entrance latency to the sucrose port becomes much faster with the CS plus than the CS minus. And so then we'll give the animals the chronic THC treatment. And then we'll do a post-test, as I said, early, first 2 days of withdrawal, late, last 2 days. And so what I'm showing you here are the things that changed relative to pre-treatment and that were the most interesting changes in this task. And the males on the left, the females on the right. And so what you can see is that the latency to enter the sucrose port after the CS plus becomes slower following chronic THC treatment. This is the green, whereas it becomes, if anything, a little faster following chronic vehicle treatment in the males. And you can see that this happens at both early and late stages of abstinence. On the other hand, in the females, we see this deficit, this delayed entry early in withdrawal or abstinence, but we don't see that late. So it's almost as if the females recover more quickly following abstinence. And you can think that, there's a possibility that this could be due to their enhanced dopamine levels and maybe the better ability to use dopamine in these tasks. But of course, we haven't shown that yet. And then the other thing we can look at is their ability to discriminate the cues. And once again, we see a sex difference and a time difference. In this case, at the end of abstinence, the males seem worse with the THC treatment. If anything, the vehicle-treated animals seem to get better. We don't see this change in the females. So especially focusing on this latency measure, there's an interesting sex difference that might be related to the dopamine changes that we saw. We don't see any changes in other measures like rewards earned or the licks per reward or the latency after the CS minus. So right now, of course, we have ongoing efforts to tie the dopamine system to this behavioral change. It involves operant changes, it involves cue discrimination, things that we think dopamine should be involved in. But in the meantime, we're developing the systems to measure dopamine in vivo while the animal performs this sort of task and more simple tasks, as I'll show you here. So, of course, as many other labs have already done, we're using the dLight engineered dopamine detector developed by Lin Tian to measure dopamine levels in the striatum in vivo. This is work I'll show you from Zhong Li in the lab. And what we're doing is a simple Pavlovian task where the animal will get a CS plus or CS minus, a much more stripped down version of the task that I showed you. And then later they'll get a reward. We use fiber photometry with fibers located in either the accumbens, the dorsomedial striatum, or the dorsolateral striatum to measure the dopamine. And as you might expect, this is in fairly well-trained animals. When we give the conditioned stimulus, the CS+, we see a large increase in dopamine in the nucleus accumbens. Also, we see a response to reward. In the DMS, in well-trained animals, we see a smallish response to the CS+, somewhat larger and fast response in the DMS to the reward delivery. And that's kind of interesting in relation to one of the talks yesterday. And then in the DLS, we see very small responses to both. And so what I'm showing you here is the time course of the development of these dopamine increases as the animal learns the task. So on early trials, of course, as the nucleus accumbens data are pretty much what you'd expect. Early trials, only a dopamine increase with reward. Mid-training, we see development of responses to CS+, but the US plus the reward, we still see a response. And then very late in training, we see only a dopamine increase to this presentation of the CS+. On the other hand, well, we also know that these are CS+ related because the CS- evokes a very small or no response. In the dorsal medial striatum, on the other hand, the fastest thing to develop is the response to the reward. It's very small at the initiation of training, becomes larger as we train the animals. And then late in training, we have a larger, tighter response to reward delivery and a small response to the CS+. But it is CS+ specific, as we don't see it with the CS-. And then in the dorsolateral striatum, first of all, I should say that this is the first time we've ever been able to measure dopamine in real time in the dorsolateral striatum. In mice, we utterly failed with fast scan cyclic voltammetry. And so what we see here are smallish responses to reward delivery early in training. They become faster and larger, middle training, and we still see them late in training. And that's the only time then we see the development of the response to the CS+. So we're still working through what these signals mean. We know they're related to reward because, as I showed you, we see responses to CS+ in reward, but not to CS+ when we omit the reward. And we see no responses to CS- in either of these regions. So we're very interested in figuring out what it is that underlies these effects. Are they purely reward related? Are they also movement related, especially in the DLS? But at least we can now get a handle on what's happening in DLS. And it's this type of data we'd like to look at with the THC treated animals in the paradigm I showed you earlier. So just to summarize this part of the talk, the female mice show increased dopamine release across dorsal striatum and accumbens early but not late THC withdrawal. Males show only one change in the DMS late in withdrawal. Females show faster recover from THC-induced deficits and operant cue discrimination tasks. So that may be related to some resilience developing, perhaps related to the dopamine changes. And then we can use this in vivo fiber photometry approach with dLight to look at dopamine measurements correlated with behavior. We've only done this with a simple Pavlovian conditioning paradigm so far, but we think it will be useful for evaluating the chronic THC effects in more complex paradigms like the cue discrimination paradigm. So I'll spend the last few slides talking about the chronic THC effects on sleep because I know people are interested in that. This is work with Karina Abrahau and Matthew Pava, along with Andrew and Alexa and Stephanie, who've kind of taken it over now. And Matthew Pava published in PLOS One a while ago that in rodents, acute CB1 activation and increases in endocannabinoids promote sleep mainly by promoting non-REM sleep. And CB1 antagonism fragments non-REM sleep, indicating roles for endocannabinoids in sleep. Sorry? Yeah, we've used polysomnography, the standard approach to measure EEG and EMG with electrodes in the mouse brain. We can separate non-REM sleep, REM sleep, and wakefulness using a fully automated scoring system developed by Matthew Pava when he was in the lab. And what we find, I'm just going to show you the data from abstinence. This is that same chronic THC paradigm that I showed you before. We will measure sleep before and after the chronic THC exposure, early and late in abstinence, and in the dark and light phases. And what it's pretty easy to see here is that looking at the change from the pre-exposure period, that in the dark phase and the light phase, we see decreased non-REM sleep in the male mice in this case. It carries over even to late abstinence in the dark phase. And it's mainly due to decreases in the duration of bouts of non-REM sleep. We don't really see much of a change in the number of non-REM bouts. So that indicates a change, a fragmentation of non-REM sleep. In the females, we really don't see much of any change at any point during abstinence or any part of the cycle, at least in non-REM sleep. So once again, the females seem to be more resilient to the effects of chronic THC. And this is something we can also explore in relation to dopamine signaling, because of course we know that dopamine is implicated in sleep. And so it'll be interesting to look at how our dopamine roles track with the THC-induced sleep disruption. We just don't have any direct connection right now, and we don't think we can explain it very well with the fast scan cyclic voltammetry data. But I will also mention that acute exposure to THC enhances non-REM sleep with a larger effect once again in male mice, and that we have this non-REM sleep fragmentation during abstinence in the male mice, but not in the females. So interesting sex differences and some compelling ideas about mechanisms that we have to look into now. So I thank you all for your attention. I'd like to thank everyone who participated, in addition to the people I already mentioned. Armando Salinas and Shana Augustine had a big hand in the dLight studies. Thank you very much.
Thanks, Dave. I see we have at least one question. Before we go to that question, I wanted to ask you a question. This is a technical question, and I know you've spent a lot of time carefully comparing dLight and Voltammetry.
Yes.
And I'm curious about the dorsolateral striatum results. So as you know well, in the dorsolateral striatum, if you take a brain slice and you locally stimulate, or if you take anesthetized animals and you stimulate, or awake animals, you stimulate a pathway, you get very robust signals. In fact, bigger signals than in ventral striatum. However, as you've seen and we've seen, the signals are often sparse during behavior.
Yeah.
And in fact, where we've seen the most robust ones in behavior in our lab has been when we have cocaine on board. So it's an uptake blocker. So we've mainly interpreted this data that there is probably a greater degree of spatial heterogeneity of dopamine release in the dorsolateral striatum, and the size of the probe is within the range that it renders it, that you're sort of hit or miss whether you're in a hot spot. It may be if you're using fiber photometry that you're observing over a broader area. Do you think that that might be something to do with the results you're getting?
That is a possibility. We know that our probe can extend in a sort of cone down about 500 microns deep from the tip, both the excitation and the collection of the fluorescence. So we do sample a pretty large area, probably larger, I would think, than with fast scan. But I think your point about cocaine is another good one. I think there's so much data because there are so many terminals in dorsal striatum that I think there's avid uptake as well. So one of the things we're trying to figure out is how much that uptake limits the responses we see during behavior. And that's something we'll be working on. I will say that in comparing the two approaches in SLICE, we know that we found, and I think Armando has shown this at several meetings along with Yolanda, is that we can detect dopamine at lower stimulus intensities with dLight and photometry and SLICE in this case than we can with fast scan cyclic voltammetry. And so we do think it is a somewhat more sensitive approach, but once again, sampling a large area versus a somewhat smaller area. And of course, in vivo, it's a big difference. We can detect much larger increases better signal to noise with dLight than we ever could with fast scan in mouse.
Yeah, and we concur that that's the same thing we find in rats, that dLight is more sensitive to behavior.
And since I love the DLS, I got to be able to measure there.
Okay, hand it over to Danny for questions.
Thank you. So we have one from the audience. I got two now, and then I also have a couple questions of my own. So first one is from Thomas Fast asking, the IP THC dose seems rather high compared to recreational or medicinal THC doses in humans, not IP, of course. How does the mouse dose compare with doses observed in humans? Are the same effects on dopamine release in mice observed with lower doses?
Okay, so I can answer the first question first. We've gone down to 1 mg per kg. We don't see any dopamine changes. We don't see any sleep changes. In fact, we see very little, if any, behavioral changes. So yeah, lower doses aren't as effective. I will say that, you know, the strong THC users will start to get the sort of levels that we'll get. Remember that mice metabolize, you know, everything faster. But they will start to get the sort of levels we can get with this injection. Granted, they're usually smoking it, so that is a different route of administration that will be interesting to look into. But the other thing is that they show very similar changes in non-REM sleep. So I think it's Ryan Vandry's work at Hopkins that has shown that. And so this fragmentation of non-REM is something that we think is at least a sort of phenocopy of what's happening in the human. And hopefully if we delve into the neurobiological mechanisms, we can start figuring out some commonalities. But it is a high dose, I admit.
And I guess going off of that is my question. For example, when human use THC is usually with terpenes, like consuming the whole plant, do you think that would have any effect if you were to administer a whole plant rather than just THC extract from the plant in terms of the?
Certainly a possibility, and these entourage effects have been touted, and there are other active agents. It's a little hard for us to do that reliably, so I don't know if we'll be doing that. What I will say is we have done CBD at pretty high doses, no change in dopamine that we could detect. And the guy who came to do that from Brazil was very disappointed because he was hoping there'd be something there.
On to the next question then. Ashela Lahiri asks, are male more or less sensitive than females to CB1 receptor dependent LTD at corticostrial synapses?
Great question. Gotta look at it. Actually, we've neglected the females there. We're finally sort of getting our act together in that regard. So that would be a great thing to look at.
And another question, maybe the last one. Do you think signals would differ if the animals were weaned off the drug rather than cut cold turkey approach?
Well, you got to remember with THC, I think they would, yeah. But you've got to remember with THC that the THC actually lingers in your system for quite a long time. That's one reason there's not a profound withdrawal from cannabis drugs. THC will stay in your fatty tissues and come back and bind to the receptors and be very slowly cleared over hours to days. So it is kind of a weaning off even when you stop THC exposure. But these are all great questions, by the way.
There's a couple more, but I don't know if we have time.
We can take one more.
All right, great. And then I guess another question from Priscilla, which there was also commented, was interesting discussion about dopamine release in DLS. Do you predict that there's more that activity in DLS versus DMS?
It's hard to say. I wouldn't have predicted so much based on, you know, my limited perusal of DAT levels. Definitely related to accumbens. But I think one thing we've seen, and I think it was mentioned yesterday, I hope I have the name right, Dr. Walden, mentioned yesterday that he seems to see faster dopamine signals in DMS than in accumbens, and he was highlighting that. And we've seen precisely that in DMS and DLS relative to accumbens. So we do think that uptake is playing a stronger role there. We're guessing that. There could be other factors. So that's something we have to look into.
Great. And I guess I just have one more question. I realize you said there might be some differences with, let's say, dopamine activity versus male and females. And do you think that could be something due to estrous cycles? Have you seen any differences there, I believe? Dr. Calipari showed that there's some differences in dopamine activity in ester cycle that is increased. So I wonder if this can apply in
certainly possible. And one thing I can say is we didn't measure that, but we saw, you know, our data in the females was relatively tight. And so if we're getting different points of the cycle, I don't think it's having a huge effect on that measure. On the sleep measure, more variability. So we want to look into that. Another thing that alcohol tends to do that I wouldn't be surprised if THC does is it also will flatten the cycle when you give high doses. So one of the things we can definitely do is see if the cycles are still intact after the chronic THC exposure, and that might explain some differences as well.
Great.
Great question.
Thank you.
Thank you, Dave.
All right, thanks.